Hello all, I Dr. Jay Bergi again welcome you all in the video series regarding diagnosis of coronavirus. Friends, this is the fourth video of our video series. In this video we will be discussing advanced testing platforms. As you are aware, we already had 3 videos regarding the diagnosis of coronavirus. In this video we will be discussing advanced testing methods. Next video we will discuss the Indian approach of testing. Now all the platforms which can be considered as the advanced platforms of coronavirus detection can be divided into the two groups. The first one is RTPCR based platforms. Now these platforms use traditional qPCR method, but some modified design aspects are there. So we will discuss the set of such platforms first, and then we will discuss few platforms which are not technically based on RT-PCR. So let's see the first platform. The most widely used platform of RT-PCR are having some design modifications in the original qPCR machines. This is a common thing. Now therefore I have kept them in the advanced method. These are not the regular qPCR machines. These are the newer version of qPCR machines. Now how they differ in compared to the traditional qPCR machine. So in compared to qPCR machines, here the PCR reaction is not carried out in either PCR tubes or in 96 well plates. So no plates are used, no tubes are used. Rather, the PCR reaction is carried out in what we call cartridges. Now, these cartridges are available as ready to use cartridges, so you need not to prepare them, it can be directly used. One. Second, these cartridges are used in the same manner by the same principle of qPCR, and therefore the complete method is known as cartridge based nucleic acid amplification test, that is CB-NAT. Now the cartridges are pre-filled with nucleotides, enzymes for the amplification, reverse transcriptase, primers and probes, and buffers. Everything is pre-filled by the manufacturer in the cartridge, and therefore the person who is doing testing has to do nothing, and the entire procedure becomes automated. Further, the detection of PCR reaction by the fluorescence detection has been miniaturized, and therefore the entire machine is made compact and portable. Now, because your machine is small, because your machine is portable, it can be installed at almost all hospitals, and this becomes what we call a point of care testing device. Just like antibody detection, now we can carry out RNA detection of virus at each hospital. So it mixes the good points of both the approaches. The RNA detection is more accurate, while the point of care testing makes it affordable and reachable to all the hospitals. The first such system and the most widely used system is Cfeed gene export system. Now as you can see, it's a cubical shape of system where there is a slot for inserting the cartridge. The blue white color cartridge you can see here. So this cartridge is placed within the system. Now the system itself will provide the thermal modulation, temperature up and down needed for the PCR reaction. The system itself will detect fluorescence, which is released as a part of qPCR reaction, and the entire system can then be attached to a PC or laptop in order to export the final result. So this is known as Cfeed gene export system. Now in order to understand how this system works, we need to see how this cartridge is working. So this is how the gene export cartridge looks like. Originally, the gene export cartridge was designed for the testing of diseases like tuberculosis. India already is using gene export cartridges for tuberculosis in its revised National Tuberculosis Control Program which is now named as National Tuberculosis Elimination Program, and we already have in country more than 1100 installations of this system with tuberculosis cartridges. Now at all these places the system is flowing, is already working, and therefore we already have trained manpower who is acquainted with the use of the system. Therefore, this particular thing, gene expert system, is very crucial in the diagnosis of coronavirus in India. Now let's see what the cartridge is made up of. So on the left hand side panel, I have dissected, opened up the entire cartridge, and as we can see here, the cartridge mainly contains of 5 different components: the lead, the processing chamber, the valve body, rotating valve body, and the foot body, 4 things. And this fifth projection outside is a transparent glass projection which is serving as amplification tube. So there are 5 major components of gene expert cartridge. Further, the central processing chamber, the main chamber itself, is having internal walls, and therefore it is further segmented in many processing chambers, each filled with different reagent, pre-filled with different reagents. So reagents, primers, enzymes are pre-filled in these chambers in the form of soluble beads. Once you add the media, once you add the liquid, then these beads will solubilize and the reagents will be active. Different buffers, wash buffers, PCR reactions will be carried out in the different section of this processing chamber. Now the sample is being added is first coming to the foot chamber, where this foot chamber, once you place the cartridge in the system, then a sonication device is there. The sonication device will break the virus structure or the bacterial cell structure in order to free the nucleic acids. So DNA or RNA will come out of bacterial cell, or in our case virus virion, by the sonication. Now once we are having liquid fluid with the virus RNA, then that has been taken up by this valve system which serves as a plunger injection, and the fluid containing RNA is taken into the central chamber. From this central chamber, with the rotation of this valve body, fluid will sequentially go in different processing chambers which will perform the purification of RNA, the RTPCR, mixing of primers, enzyme and everything. And once everything is mixed, the fluid will come in this amplification tube, which is now inside the gene expert system, where the temperature up and down will be carried out and the RT-PCR reaction will take place. Now because this thing is transparent, the fluorescence released by the probe will be detected by the gene expert system. So entire cartridge works as a self-contained system. We need not to do anything, just sample is to be added. Cartridge will carry out RT-PCR and qPCR at its own, and no reagents are to be added. A fully automated, fully self-contained system. Now the reagents, that is disease specific, virus specific probe or primers, are there in the form of powdered beads in the cartridge itself. So if you want to diagnose coronavirus, then the only thing that manufacturer needed to do is change those primers which were pre-filled in the cartridge. The machine or the system which is already installed in more than 1000 laboratories in India for the TB need not to change, and that's what company did. They came up with a cartridge specific for SARS coronavirus 2. So now the same gene expert system which we were having for the tuberculosis across laboratories in India can be used for SARS coronavirus 2 diagnosis. ICMR has also published guidelines for using this and considered the use of this system validated for the purpose of coronavirus diagnosis. A similar system is manufactured by a Singapore based company known as Credo. This is Vita PCR system of Credo, where a oropharynx swab collection is carried out. Then the swab is mixed in a specific tube provided which contains the lysis solution. So the RNA is lysed, RNA comes out into the fluid, and then the RNA containing fluid is transferred using pipette to a specific tube which is pre-filled with all the PCR reagents. This tube is then placed into the system where the RT-PCR reaction is carried out and the reading is taken. This takes around 20 minutes. Reading is then given on the screen itself. So a very compact system of qPCR by Credo Vita PCR system. The same principle of qPCR is carried out here. A third system is been designed by a German company known as Bosch. Again, there is a central hub, a central system. Then there are cartridges which are pre-filled with all PCR reagents needed. How this thing works? So first, there is a laser reader in the central hub. The laser reader will read barcode, thereby we identify the specimen of the patient. The same reader will read the cartridge barcode and will attach and will assign cartridge to the individual patient. Then in the cartridge opening, we need to transfer the transport media containing virus, and then the cartridge is to be placed into the system. RNA purification, amplification by PCR, and the reading of fluorescence, everything is done by the same central hub. So this is the machine by Bosch. The miniaturization of PCR is then going ahead by a company called Biotech. The system is known as Aquila system. Here the machine is so miniaturized that it can be handheld. Instead of calling it a system, now it is known as dock, something where you can dock the cartridge. So how it works? Similar process. There is a lysis solution provided. We need to mix our swab into the lysis solution. So virus present in swab comes to the lysis solution. The outer layers of virus are lysed. RNA is released. There is a specific pipette which will deliver purified RNA to the cartridge. The cartridge contains pre-filled reagents, probes, primers, nucleotides for the qPCR reaction, which is then been read by the dock, and the dock will give you the result. Fortunately, the similar system is been already there, been designed and been used in India also. So there is a company called Mole Biodiagnostic. It's a Goa based company which is already having a system known as TrueNet system. Now just like the Gene Expert system, TrueNet system is also been used in India for the tuberculosis diagnosis. But it has got an advantage of being indigenous. Being of Indian origin, the system is very cheap and convenient to use. The system contains two different machines which are available in a single carry case. The first machine is for the RNA or DNA extraction and purification. So we need to place sample here. The DNA or RNA is purified and extracted here. Purified RNA or DNA is then delivered into a qPCR system where the PCR reaction is carried out and the result is being read. The entire system is battery operated. Reagents are not to be stored in refrigerated condition, and therefore this system is suitable for the Indian use. Now the reagents in the TrueNet system are placed in a what is known as smart chip. So the TrueNet smart chip looks something like this. Reagents or what we call master mix is already present in a microfluidic chip. From this, reagents come to a very small thermal cycler where we are adding sample also, that is purified RNA or DNA, and then the qPCR is carried out and reading is taken. So this is what we call the TrueNet chip. Being indigenous system, the advantage is that this system is very cheap. Now here in Chhattisgarh COVID laboratory, there are 11 such systems been installed. So you can have multiple systems, we can afford having multiple systems which are fully automated doing RNA extraction and qPCR in a small laboratory of Chhattisgarh. Now there are other molecular biology systems. Now remember, in all these advanced diagnostic systems, we are only discussing the direct approach diagnostic method, that is the method that only detects the virus RNA. We are not discussing antibody based system. All the platforms that we have discussed by now were based on the RT-PCR and qPCR. But there are some platforms available which do not use qPCR. Let's see such thing. One of the most interesting device is by GeneMark and is known as Plex system. Here there is a central hub which will control the side hub, and this hub will have cartridge slots. Now here you can see 3 cartridge slots, but you can have a hub with the 5 cartridge slot. You can even attach an extra hub with more 5 cartridge slots. So the capacity of the central hub can be increased by the sub hubs. The principle upon which this thing works is very interesting. The process is really simple. First, we need to transfer the transport medium containing virus to the cartridge. Then a barcode scanner will read the cartridge. Then the cartridge is to be placed in the side hub. The central hub will carry out the reaction and the report is generated. So fully automated self-contained system again. But the principle is unique. In this case, the cartridge is having gold plated electrodes in the bottom, and these gold plated electrodes in the bottom will have an immobilized DNA, single stranded DNA probe. Now this sequence of this DNA is complementary to the sequence of the virus that we are detecting. So in our case, the DNA which is immobilized on the golden electrode is having sequence complementary to the specific site or the specific genes of SARS coronavirus 2. Then there is a microfluidic device in which a single stranded nucleic acid complementary to this immobilized nucleic acid with probe is added and the patient's sample is added. Remember, the patient's sample will have purified RNA which is not having any probe, but our system itself is having a purified nucleic acid which is having probe. Now these two will compete to bind with this immobilized RNA. The amount of probe containing system nucleic acid which binds to this immobilized RNA will depend upon the competitive RNA present in the sample. So if the sample is having more virus, the probe will bind to the immobilized DNA less. If the sample has got no RNA, more probe will be binding here. Now what is the probe? So probe here interestingly is not a fluorescent dye. Rather, it's an organic compound known as ferrocene. Now the ferrocene is having conductivity property, and if it is bound with the electrode, then there will be more electrode current. If it is not bound with the electrode, then there will be less current. So ultimately the binding of nucleic acid to the specific sequence present is detected by electrochemical reaction, and there is no PCR reaction carried out. We are just measuring the current which is passing in this electrode. Although the current is very weak, it is microamperes, so a very weak current is generated and that is being detected, and thus the presence of nucleic acid in sample can be detected. This is known as GeneMark Plex system. There is one more interesting system known as Microsense RepiPrep. Now Microsense RepiPrep is also using cartridges, and these cartridges are coin-like disc. Within this cone-like disc there is an opening from where you need to add sample, and then these cartridges are placed in this very small pH meter like machine where the reaction is carried out and the result is displayed. Now reaction carried out here is not PCR, but it is very much like PCR. Just like PCR, the nucleic acid amplification is there, but it is not through the temperature up and down. There is a new way of nucleic acid amplification known as loop mediated isothermal amplification. It's a different thing than PCR. Here again, the sample RNA or DNA is increased in the concentration by the amplification using primers, but the primers once synthesize the new DNA or RNA, that product is looped by itself due to the presence of internally complementary sequences within the primer. So we design primer in such a way that it has got complementary sequence then to the sequence of our target RNA or DNA. So due to the complementary sequence, the ends of primers are looped. Then using a second set of primer, the amplification will be carried out again. But the amplification cycles do not require dissociation of double stranded template. So you need not to have temperature increase, need not to have temperature decrease. There is no variation in the temperature, so no thermal cycler is needed. Amplification is there, but it is based on loop. So this is known as loop mediated amplification, carried out at the constant temperature, so isothermal. This process is carried out by the Microsense RP prep, and therefore it is very much miniaturized, again detecting the specific gene of coronavirus. There is one more system doing the same isothermal DNA amplification that has been developed by a company called Abbott, a very well-known company of US. Now here cartridge is available into the three components: a sample receiver, a transfer cartridge, and the test base. Now the test base already has got two test tubes which are having dry powdered reagents needed for the reaction. The patient's swab is transferring the viruses to the sample receiver. From sample receiver, purified nucleic acid is transferred using transfer cartridge to the test reaction. Everything goes into the machine here, which will provide constant temperature for the isothermal amplification. The result is recorded here. So a very complex system doing isothermal amplification of specific virus DNA. What is the procedure? So the sample receiver has got a cover. We need to remove that cover. Then the swab having virus is mixed with the sample receiver. Then the purified RNA by the virus breakage, lysis solution is already there in sample receiver. So the lysed virus sample with RNA is then transferred to the test reaction using this transfer chamber. It goes into the test chamber. The test chamber is having test tube beneath. Everything goes into the machine and that will carry out the reading. Remember, the process is, or the principle is, isothermal amplification. The entire thing can be carried out in the span of 10 minutes. So within 10 minutes, we'll be getting result of whether the specific virus sequences are present or not. This is the system by Abbott. There are tests being developed which are not based on any of such DNA amplification. These are the tests which are based off a gene editing technique known as CRISPR. We are all aware that CRISPR-Cas9 is a revolutionary DNA editing tool, but the same DNA editing enzymes can be used using specific probes to identify sequences present in the sample. So the particular gene in the virus RNA can be identified using CRISPR, and the entire thing is so designed that it is being carried out in a lateral flow test strip. So it's a strip test, and therefore it doesn't need any machine. This thing is been developed in two of the US universities and one of the CSIR research laboratory in India. However, they are still in testing phase, and in market this test is not available yet. So it is truly futuristic test. The advantage is that no sophisticated machine is needed. Disadvantage is that because there is no amplification, the copies per microliter of sample, the amount of RNA per microliter of sample which can be tested is limited. So you can only detect when the virus load is higher than a certain level. This is what we call CRISPR based strip test. Lastly, a rare new approach has been suggested. This has just been suggested in the research paper, not available in market. In this method, the different samples are placed in microwave plate, 96 microwave plate, where a specific probe has been added. The probe is created by the machine, which is having specific barcode signals specific for each sample. Now because each sample has got a unique sequence of barcode, we are knowing which sample is been having which barcode. Then reverse transcription is carried out. After reverse transcription, the entire sample in 96 well plate is mixed in a single test tube. Then the PCR is carried out, and then the product of PCR, which is the mixture of all these 96 samples, will undergo what we call next generation sequencing. The next generation sequencing will identify each nucleic acid template available here, and the complete sequence that we will identify includes the barcode. So in the final result, we know that which barcode, that means which patient's sample is positive, and which barcode, that means which patient's sample is negative. The advantage of this system is that we can run many samples at one go. As many as 19,000 samples can be tested in just one run of next generation sequencing. The disadvantage is that it requires an NGS platform, which is again expensive. Now again, this is again not available currently in market. It's a futuristic test. So these are all the tests which are going to change the scenario of coronavirus detection in coming months. Thank you all.